Right, grade 12. So now we are busy winding up. We're finishing off our protein synthesis. Remembering how important those proteins are in the body. I mean, if we look at it, not only are they structural, keratin, myosin, they are also our hormones. So they are in control of our chemical controlling of our body. They are our antibodies, so they're our fighters in the body. And that is just to mention a few of the functions of the proteins. So we have been talking right through this section on the code of life. We've been talking about the genetic code. And so this little unit is just going to sum up for you what we actually mean by the genetic code. And so it's going to include quite a lot of what I have said before. And then I'm going to throw in a little section on mutations, which we will pick up again when we do genetics. But it links directly to what is found on the DNA. So I felt that we really have to make mention of it at this point. So when we have a look at the genetic code, the genetic information obviously is stored in the chromosomes in structures that we know as being genes. So our original genetic code came from the fusion of dad's sperm and mom's egg and that zygote that is formed. And then our cell started to copy and divide, as we had a look at in grade 10 with mitosis, and as I mentioned to you when we did DNA replication. So those genes have to be copied identically into the trillions and trillions of cells that make up your body. So when we are having a look at protein synthesis, the code is on the gene, so it's on the DNA, and then that code is expressed in the messenger RNA through transcription. So the code is transcribed onto the messenger RNA, copied onto the messenger RNA, and then that messenger RNA goes out to the ribosome to form the different proteins. So the DNA is our code. It is transcribed, it's copied to the messenger RNA, and then the ribosome translates it into a polypeptide or into a protein. So it starts with our double-stranded DNA. You should be really like, I know what that is, double-strand thiamine, it's DNA, yay! A section of it is known as a gene. They often describe a chromosome as being a linear arrangement of genes. Linear, a line, gene on top of gene on top of gene on top of gene. So your genetic code is made up of a sequence of bases. So again, we could say that what we are looking at in this diagram with our 4 different pairs of nucleotides could perhaps be the gene for, let's say, for keratin. Okay, so that is the gene for making the protein that makes up our hair. Okay, so when we have a look here, here is hemoglobin. Now to remind you, hemoglobin is your red pigment which is found in your red blood cells, and its function is to transport oxygen. And we need oxygen in our bodies because you can eat as much food as you want until that oxidation of the glucose has taken place during cellular respiration. You're not going to have the chemical energy you need to be doing what you are doing all the time, and in fact for your body to be functioning as it does. So hemoglobin is a protein. It's actually, that globin is the protein, and then it's got iron joined onto it. And so the body needs to be producing this hemoglobin all the time. And so how does the body know how to produce this protein? Well, there is a gene on the DNA which codes for the making of this protein. That gene, that message is transcribed onto messenger RNA, and then my ribosome actually brings those amino acids together with the help, thank you, of transfer RNA. So here you can see is the genetic code for hemoglobin. How do I know it's the DNA? Because it has thiamine, Why is there only one strand? Because only one strand acts as a template when proteins are made. And here is my messenger RNA. And these bases are complementary. So these are showing me the amino acids that are brought into the chain while hemoglobin is being formed. So that whole genetic code, the gene, the message on how to do this came from DNA. That's why we talk about DNA having our genetic blueprint for who we are in our 46 chromosomes. Have a look at this. Remember I told you that a transfer RNA, a messenger RNA, only codes for a specific type of amino acid. So here, very nicely shown, you can see two amino acids which are the same, their messenger RNA are the same, and of course the message on the DNA is the same. So we transcribe from DNA to messenger RNA, and that will ensure that the correct amino acids are brought into the correct order. Okay, so remember that there are three processes here. Replication, because you only start off life as a single cell. DNA makes an exact copy of itself. DNA contains the genes, the genetic code. The genetic code, there it is transcribed onto the messenger RNA, shown very, very nicely for you there with your two different colors, your double-stranded DNA, your single-stranded messenger RNA. Of course, messenger RNA moves out of the nucleus, so there it is, it's on the move, off it goes, and it goes to the ribosome. So this beautiful yellow organelle over here is the ribosome, and that is where translation happens. So we've got replication, transcription, translation, and not one of you are going to go into the exam without being able to describe and explain those different processes. On the ribosome, my protein is formed, my amino acids in a specific order to make a specific protein. So when you talk about transcription, you don't say a protein is made, you say a specific protein is made. So there we've got DNA transcribes to RNA, and RNA translates to a protein. DNA contains the genetic code. So that's where our word genetic code comes into the whole process. So now when we have a look at our genetic code, when replication happens, sometimes there is a copying mistake made. So when my DNA makes an exact copy of itself, sometimes a mistake is made. And when we do mutations in genetics, I'll actually point out to you it actually happens a lot more times than we are even aware of. But today we are talking about a mutation that changes a protein. So when we have a look at that word mutation, mutation means a sudden change in the nucleotide sequence. Okay? Again, when we do genetics, we will have a look at how mutations happen. So if I change the sequence in my nucleotides, it is obviously going to bring in different amino acids. So let's just take a very simple example here and say, this is my, this is my original DNA. C, 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 let's give it a G, an A, and a T, and an A, and an A, and a T. So that will bring in specific amino acids. This one's going to bring in the square, this one's going to bring in the triangle, this one's going to bring in the star. And they are going to bond together, and that forms my growth hormone, which allows my muscles and my skeleton to grow at different periods of my life. Now there is a mutation, a change, a copying mistake happened. And so This one was fine, it didn't change. But this one, there was a mistake in that particular nucleotide. And this one stayed the same. What amino acid is going to come in here? The square. Is the triangle going to come in here? No, because my bases have changed. And so now a different amino acid comes in here and the same one comes in over there. Is that protein going to have the same structure? No. And that may affect its function. And so that we call a mutation. And that is how mutations link into protein synthesis. So if we have a look at this visual over here, this is the normal sequence of hemoglobin. That's the abbreviation for hemoglobin. Now a mutation happened. Okay, so if you look at this, that and that is the same. There was no copying mistake. That and that is the same. But over here, instead of us having CTC, we've got CAC. So will glutamic acid come in here? No. A different amino acid will come in at that place. And that means that my protein may have a different function to it. And I am stressing the word may because many mutations actually don't affect the functioning of the protein, but it just might affect the functioning, which means that hemoglobin is not going to pick up oxygen in the same way, and that person could then suffer from a disease like anemia. And so that is how mutations affect protein synthesis. So you can see here that instead of us having threonine, proline, glutamic acid, and glutamic acid again, we've got threonine, proline, valine, and glutamic acid. Those three, and it is on the messenger RNA, so of course we are going to call that a codon, will bring in a different amino acid. So they could give you a question on protein synthesis, and then they could throw in a mutation question by changing the order of the bases. And so you would need to be able to link our protein synthesis to the base sequence, which comes originally from the DNA. So I hope that, and of course that is pushing your level of questioning up to maybe a 3 or a 4 level question, and we want to be able to get those questions right as well as our level 1 and 2 questions. So grade 12s, just bear that in mind, mutations. We will talk about it again when we do genetics.